Okay, next up we have Laurie Passmore. Laurie is a group head at the MRC Laboratory of Molecular Biology. She studied and trained at the universities of British Columbia and at the ICR in London, and then she joined the LMB as a postdoc, where she stayed afterwards to start her own lab back in 2009. So I guess she's a bit of an LMB lifer now, sort of, and then it's working on gene expression, RNA processing, and DNA repair. Laurie, it's all yours.
Great, thank you. Hopefully, can you see the screen fine?
Yeah, looks good.
Good. Thank you, Alan, for the introduction, and thank you, Julia and Chris and everyone else for organizing this. It's been great so far, and looking forward to today and tomorrow. So my lab uses cryo-EM alongside other structural and biochemical techniques to understand protein complexes that interact with DNA or RNA. And so today I want to discuss some of our work that aims to understand the molecular mechanisms of DNA crosslink repair. So DNA crosslinks can occur after exposure to chemicals, including chemotherapeutic drugs and alcohol, but also as a result of just normal cellular metabolism. So these crosslinks block DNA replication and transcription, and if they cannot be repaired, it results in genome instability, leading to human disease, including Fanconi anemia, or FA. So FA patients have a wide spectrum of symptoms, including developmental defects, bone marrow failure, and a predisposition to cancer. But at the molecular level, all FA patients have the same inability to repair DNA crosslinks effectively. So this FA DNA repair pathway involves a large multi-subunit E3 ubiquitin ligase called the FA core complex. So in response to DNA crosslinks, the FA core complex monoubiquitinates its substrate proteins called D2I, and this in turn recruits nucleases that allow the removal of the crosslink and the repair of DNA. So like many E3 ligases, the FA core complex has a ring finger subunit, and this is located in FANC-L. So a number of years ago, it was shown that if you just purify the FANC-L ring protein on its own, it's able to mediate the site-specific monoubiquitination of FANC-D2. However, mutation of any one of the core complex components is thought to abolish ubiquitination and leading to defective DNA repair and disease. So a major question really was then, what are the roles of the other FANC proteins within the FA core complex? Now, most of the subunits in this complex do not have much similarity to proteins of known structure. So we couldn't really infer the roles of the proteins simply from the sequence. And there weren't many crystal structures of the individual subunits. So for many years, an understanding of how this entire pathway worked was really hampered by the lack of a fully purified in vitro system. And this is because there were a number of challenges. So first of all, it's pretty low abundant in cells. It's not found in lower eukaryotes, so we couldn't, for example, purify it from yeast like we often do. And of course, its complexity and size generated additional challenges. So about 10 years ago, we decided that we would purify the native FA core complex from the chicken DT40 cell line with the aim of imaging it using cryo-EM. So this is work done by a postdoc in my lab at the time, Isan Rajendra, and we worked in collaboration with KJ Patel, who was also at the LMB. So Isan was able to purify the entire complex from chicken cells, and he showed that it's comprised of 8 stably associated subunits. He also reconstituted its biochemical activity in vitro and showed that the intact complex was more active than the ring subunit alone. He showed that efficient ubiquitination required the presence of DNA, and also that 3 proteins, B, L, and 100, form the catalytic core of the complex. However, this is a silver stain gel, and really the quantity and purity of this protein was not sufficient for structural investigation. So we decided that to move forward, we needed to make a recombinant complex. And so this is work that was started by Eason, but then continued by Xabi Shakil and Pablo Alcon. And so what they did was they inserted all 8 genes for the FA core complex into a single baculovirus, so that when they infected the insect cells, all the subunits are simultaneously overproduced and the insect cells produce the complex. And so ultimately, this allowed them to obtain the complex in high purity and in milligram quantities for the first time. Now, this purified complex is active because it adds only a single ubiquitin onto the FANCD2 substrate, and it does this only in the presence of DNA and the FANC-I protein. So the first thing we did, of course, was we took this purified complex and we imaged it using cryo-EM. And we obtained micrographs that look like this. So I know these look a little messy, but they do contain particles. And here I've circled some of them in yellow. And when we performed 2D class averaging, the 2D classes looked pretty good. So we could see really nice secondary structure, lots of details, especially at the center of the particle. For example, here in yellow, I circle a coiled coil, which is at the center of the complex. So using these data now, we were able to obtain a 3D reconstruction. So the structure is around 25 nanometers in length. It's pretty extended, and the overall resolution was around 4.2 angstroms. However, the periphery, as you can see from the map here, is much more poorly resolved than the central core. And we suspected that this was due to conformational flexibility. So we analyzed this computationally using multi-body refinement in Relion. And this showed really a continuum of conformational movement, where the FA core complex both twists and bends. But fortunately, we could then use particle subtraction followed by focused classification and refinement to reconstruct 3 separate maps. So we separately made a map for the top of the complex, shown here in yellow, the middle, shown here in blue, and the base in pink. And now there was really sufficient resolution to dock any existing structures, and we could clearly resolve secondary structure elements throughout the map. However, as I've already mentioned, there weren't many existing structures. So these only accounted for about 12% of the entire mass of the complex. So therefore, we really needed additional information to figure out where each of the FANC proteins were located inside this structure. So the first thing we did was to assign subunits to specific regions of the map. We purified and imaged some subcomplexes using cryo-EM. And so by comparing the 2D classes of these subcomplexes to those of the complete FA core complex, we could identify regions that were missing and therefore corresponded to the missing subunits. So first, if we removed FANC-A, the largest subunit in the complex, nothing really changed in the class averages. So this really suggested that FANC-A is either conformationally heterogeneous and blurred out in the maps, or that it denatures and dissociates during cryo-EM specimen preparation. Second, if we image a complex containing AGBLN100, we miss the base of the complex, circled here in green. So therefore, we think that this base in green contains the CE and F subunits that are missing from those complexes. And then circled here in blue is a partially disordered arm that extends out from the central part of the complex. And we think this is FANC-G because this density is lost whenever FANC-G is removed from the complex. So we could use this kind of dropout experiments to really locate where the subunits were in the map. We also used native mass spectrometry in collaboration with Carol Robinson and George Cherov in Oxford to determine the subunit stoichiometry and arrangement. So here, the largest complex that we detected is just over 800 kilodaltons. And this contains 7 of the 8 different subunits of the FA core complex, including 2 copies of the BLN100 subunits and a single copy of the remaining subunits. So this was really strong evidence that there wasn't a unit stoichiometry for each of the subunits. Next, we determined which residues are in close proximity using cross-linking mass spec with Francis O'Reilly and Yuri Rapsilber in Berlin. And so they identified 834 high confidence inter and intramolecular crosslinks. And so now by combining all of this information, we could place secondary structures and build backbone models. However, because we didn't really have good homology models for most of the subunits, it was still really difficult to derive meaningful models at this resolution. So to improve our models, we collaborated with Frank DeMaio and Dan Farrell from his lab. And they used a method that integrates TIA Rosetta and EM map guided complex assembly. And the pipeline for that method is shown here. So first, shown on the left, they generate multiple sequence alignments of the subunits and use these to manually segment the proteins into different domains. Next, they used TIA Rosetta. And this uses a convolutional neural network to predict both the distance and the orientation between pairs of residues in a protein. And then it uses these data to constrain folding in a fast model building protocol. Next, Dan used an automated search of the cryo-EM reconstruction for the best matching placements for each domain model. And by using Monte Carlo sampling of the domain assignments, he could find the arrangement of domains that maximizes the agreement with both the cryo-EM and cross-linking mass spec data. And then finally, Dan rebuilt the connections between the domains and refined the entire complex against the cryo-EM map. So in total, Dan used 42 different deep learning guided models for the FA core complex subunits and docked these into the cryo-EM density in a way that was maximally consistent with the experimental data. So ultimately, this allowed him to place almost 5,200 residues, which is now over 80% of the complex. And so we now have models for 7 of the 8 subunits. We found that in the center of the complex here, there are 2 copies of the catalytic core. So that's the BLN100 subunits. And arranged around this are all the other subunits. So there's 1 copy of each of the green subunits in the base and 2 copies of FANC-G on the side. I'm just going to talk about a couple of details of the structure. So if we look in more detail at the BN100 subunits, we see that they're not only part of the catalytic module, but they also form a molecular scaffold that really coordinates the positioning of the ring subunit and all the other proteins within the complex. So there's 1 of each subunit in the bottom half and 1 of each subunit in the top half. So really the core of the complex is a dimer of heterodimers. Now, each of the proteins, FANC-B and FAP100, start at the outer part of the complex with a beta propeller. And then this is connected to the central structure by long intermolecular coiled coils. Now, this dimer of heterodimers is not symmetric. And that's because the other subunits of the complex alter the positions of the domains and break the symmetry. However, we think that this complex would be symmetric in the absence of the other subunits, as shown in a negative stain EM study by Alessandro Costa's lab. Now, although these 2 proteins do not have similarity to each other at the sequence level, they looked almost identical in the structure, and this was a big surprise to us. So they both start with a beta propeller, and this is followed by an intermolecular coiled coil, a mixed alpha-beta central domain, and then a helical bundle at the C terminus. So this really suggests that these 2 proteins may have a common evolutionary origin. Now, what we really wanted to find out was how the ring subunit FANC-L was placed in the map. Now, as I already mentioned, there are 2 copies of FANC-L, and FANC-L has 3 different domains. So a model of the full-length protein based on a previous crystal structure from Helen Walden's lab fits into the base of the complex. And this is really cradled by all the other subunits in the base. In contrast, we could only visualize one domain in the second copy of FANC-L, and this one's at the top of the complex. So the ring was not visible. And we think this is because it's flexible and it's therefore blurred out in the structures. Now, because there are two copies of FANC-L and that each have different structures, we hypothesize that they must play different functional roles in the complex. And so here I'll summarize what we've learned by using this cartoon of the structure of the FA core complex. So FANC-B and FAP100 act as molecular scaffolds to really coordinate the positioning of FANC-L and all the other subunits of the complex. This really explains, I think, why the intact complex is more active than isolated FANC-L, because the other subunits are required to stabilize the fold of FANC-L. So FANC-L in the base is fully ordered, whereas the FANC-L in the top is flexible. So we hypothesize that the one in the base is really just playing a structural role to help scaffold the other subunits. Whereas on the other hand, the one on the top is accessible and available to bind to an activated E2. So together, this would really optimally position substrate in E2 to allow ubiquitin transfer. And this hypothesis was confirmed by a more recent structure of the FA core complex bound to both E2 and substrate from the Pavlovich lab. And that was just published just recently in NSMB. So what does this tell us about disease? Well, most of the FA patient mutations are found in FANC-A, which is shown here in blue. However, FANC-A we think is flexible and it's not well ordered in our map. Now, some patient mutations are in the substrate recognition module in green, but the mutations are really rarely found in FANC-B or FAP100 or any of the catalytic core. So when I map the patient mutations on the structure shown here on the left, you can see that most of the mutations are really in the structural periphery of the complex. So we think that the structure is telling us that this is because disruption of any of the central subunits would destabilize the entire FA core complex, resulting in a non-functional FA pathway that may be incompatible with viability. So in agreement with this, the few patients who do have mutations in the central core of the complex are generally much more severely afflicted. So overall, this structure of the FA core complex gave us insight into how the complex functions as an E3 ligase and provided insight into the mechanism of disease. But really a long-standing mystery was why monoubiquitination and deubiquitination is so fundamental for cross-link repair. And so over the last few minutes, I'm just going to tell you a little bit about our work on the substrates. So an X-ray crystal structure of the FANC-D2 FANC-I dimer was determined almost 10 years ago by the Pavlovich lab. And the structure is shown here with FANC-D2 in blue and FANC-I in pink. So this showed that the subunits are really similar to each other, and they have a shape of a saxophone. And the two subunits associate with each other via a back-to-back mechanism. Now, what was intriguing about this structure is that the ubiquitination sites on both subunits map to the dimerization interface. So they're really buried in between the two subunits, really raising questions about how the FA core complex can access and ubiquitinate these lysines, given that they're buried at the dimer interface. Second, it was really unclear why DNA is required for monoubiquitination. So to address this, we used our in vitro reconstituted system to purify and image both the unmodified and ubiquitinated substrate proteins. So first, the structure of the D2I complex without ubiquitin is almost identical to the crystal structure I already showed you. So again, I've colored D2 in blue and FANC-I in pink. And although this sample was prepared in the presence of DNA, DNA is not visible in the map. We know that it does bind DNA, but we think it must have a fast off rate. We next determined the structure of the ubiquitinated complex at 3.8 angstroms resolution. So it was immediately apparent that major conformational changes had taken place. So ubiquitin is in green, and it was located at the back of the complex, wedged in between the two proteins. And the C termini of the two subunits had folded inwards and contact each other via a new interface. Now, DNA is required for ubiquitination. And as I already said, we included DNA in both of these samples, but it was only visible in the ubiquitinated complex, and it's shown here in yellow. So since this complex is really wrapped around DNA, these structures show that the D2I complex is a DNA clamp. And so let me just show you a couple of details of the structure. So the DNA binding channel contained density for around 15 to 20 base pairs of double-stranded DNA, but the DNA kind of extended beyond the channel, so we could model a total of 33 base pairs. The DNA is at a lower resolution than the rest of the map, likely because there's heterogeneity in both the position of the DNA as well as some flexibility. And the repositioned C-terminal domains of D2 and I really contact each other with these alpha helices, as shown here, over top of the DNA. Now, ubiquitin, on the other hand, is located on the opposite side of the complex, and here it's colored in green. So the C-terminus of ubiquitin is covalently anchored onto a lysine side chain of FANC-D2, but it really docks onto the surface of FANC-I. And we think this probably stabilizes the complex, locking it into this conformation. So again, you could really see that even though it's linked to the blue D2 protein, it reaches across and sits on top of the pink FANC-I protein. And in fact, there's really a lot of contact between FANC-I and ubiquitin. Now, if I superimpose both the unmodified and the ubiquitinated models, you can see how the dimer interface really acts like a hinge, allowing the C termini of the two proteins to swing towards each other. So there's a rotation of almost 70 degrees in the monoubiquitinated complex, allowing it to clamp onto DNA. Now, this is really largely a rigid body movement of both subunits, but the N and C termini are also remodeled. So if we look more closely at the DNA, you can see there's a pronounced kink of about 60 degrees in the double-stranded helix within the complex. And so we don't understand exactly what this means, but it may be involved in recognition of the crosslink, or it may prepare the cross-linked DNA for repair. So how do the E2 and the FA core complex access the lysine for ubiquitination? Well, DNA binding opens a cleft, exposing the ubiquitination site on the backside of FANC-D2, as shown here on the left. So interestingly, it's really DNA binding that closes the complex and exposes the ubiquitination site on the backside. And you can really see this quite clearly from the end-on view. So together, our structure and biochemistry explains why and how DNA stimulates FANC-D2 monoubiquitination. So the complex binds to a double-stranded DNA, promoting closure of the heterodimer, exposing the ubiquitination site on the backside of the complex, and allowing access to the activated E2 and E3. So we also hypothesized that ubiquitin acts as a covalent molecular pin, locking the closed heterodimer on DNA and preventing it from rotating back to the open D2I conformation. And so finally, a major remaining question was that given that these two subunits are so similar, why is a heterodimer really required? And so to address this, we purified the two proteins separately. And I'm not going to show you all the data, but we found that FANC-I is a monomer and it binds DNA. FANC-D2 is a dimer and doesn't bind DNA, and neither isolated subunit is ubiquitinated. So to understand the molecular basis of this FANC-D2 dimerization and gain insight into why it doesn't bind DNA, we determined the structure using cryo-EM. So this FANC-D2 dimer is symmetric, with each protomer containing the same overall architecture as in the heterodimeric complex. But surprisingly, this D2 dimer was in a closed conformation. So it looked more similar to the ubiquitinated complex rather than the unmodified complex. So this provides a molecular explanation for the lack of DNA binding by the isolated FANCD2. So the DNA binding sites are blocked because it's in this closed conformation that presumably does not open to allow entry of the double-stranded DNA helix. Now, in addition, in the homodimer, the lysine residues on the back of the complex aren't accessible, and this really prevents their ubiquitination. So this suggests that there could be a mechanism of autoinhibition whereby isolated FANCD2 can't be prematurely recruited to DNA and can't be ubiquitinated prior to assembly with the FANC-I subunit. So this is my last slide. And just to summarize our model, which is that the isolated FANCD2 protein forms a homodimer that can't be ubiquitinated and doesn't bind DNA. I didn't show you, but FANC-I can displace one of the protomers, forming an open complex that can bind DNA. The complex binds DNA at stalled replication forks, and this causes this hinge-like rotation of D2 and I, so they encircle double-stranded DNA. This exposes the ubiquitination site, allowing the FA core complex and the E2 to access the lysine. And ubiquitin locks this DNA clamp onto DNA. This complex can then recruit the nucleases to repair the DNA crosslink. And because ubiquitin locks the complex on DNA, ubiquitin really must be removed in order for the clamp to open and to release the DNA. So I think I've thanked most of the people as we went along. I should say we also worked with Sheraz, who implemented a local symmetry algorithm in Relion that for averaging the two halves of a subcomplex map that I didn't discuss here. And of course, I'd like to thank lots of people who were involved and also EBIC for help with data collection. And I'm happy to take questions.
Thanks, Laurie. That was another great talk. I've got quite a few questions, so we better get started because we're running out of time. So Katsu Murakami has said, fantastic structure and talk. Did you treat the FA complex with cross-linking or graphics or anything before to stabilize it?
No, we didn't have to use any cross-linking. It seemed pretty stable on its own.
Abid Javed asks, in the D2I closure complex upon binding, is the double-stranded DNA fixed, or is there some coordinated flexibility within the DNA as well?
So we don't really know exactly. So one of the things we don't understand is what gets this complex to the right place. How does it get to the crosslink? So we don't understand whether, maybe it closes on DNA, slides along and finds the damage, or does it actually only get ubiquitinated at the site of damage? There's lots of questions that we still don't understand at exactly what happens before and after the ubiquitination and closure of the complex. So yeah, good question. We don't understand.
Okay. Julia Zanetti has asked, you mentioned that in the dimer within the full complex, just one subunit is active and the other one's just structural. Do you think that they cycle and switch?
We don't think so, because the one in the base is really buried and surrounded by all these other subunits. Of course, there could be some major conformational changes, but the structures from the Pavlitic lab suggest that's not the case, and that, I guess maybe there's been some duplications at some point, and one of them just became simply structural. So the E2 binding surface is buried, and we don't think it would be active.
Okay. I'll quickly squeeze in one or two more. So Youngjin Kim says, great talk. I don't think you talked about this, so maybe he knows it from your paper. Why do you collect D2I data from different tilt angles?
So it had a preferred orientation. And so to fill in some of the missing views, we collected a bit of data at 23 degrees.
Okay. And then there's a few more on the chat. Laurie, I hope you can fill them in. I'm going to squeeze one in myself, actually, just before, as to use my position as chair. So for the integrative modeling, did you have many alternative models to search through? when you combine it with this whole machine learning method plus all these different fittings and everything, was there a lot of different candidates to search through, or was it quite unambiguous?
Yeah, I think that they usually looked at the top 3 models, and in some ways, because they didn't use the cryo-EM map in making the models. Right. So usually always the top model, the top scoring model fit best in the map. So the map kind of acted as a validation in some way of the model. Yeah. So if you made models and then that model fit really well in the map, it was somewhat validation of the model. Sure. So yeah, they tested the top 3, and in almost all cases it worked. In a few cases there was some tricky bits and they had to do a little bit more manual modeling and fitting, but more or less it did seem to work fairly well.
Okay, great. Okay, thanks very much, Laurie.
Thank you.
Move on.